My name is Natalia Arai. I am a Hostos Community College liberal arts student. My mentor is Professor Chowdhury, and our research topic is autologous stem cell transplantation and regenerative treatment of type 1 diabetes. Research background. Type 1 diabetes is when the body has an autoimmune deficiency that kill the beta cells that are produced by the pancreas. These beta cells are really important because they're going to regulate the blood sugar levels in our system. It's often called childhood onset diabetes or juvenile diabetes, and there's no prevention for it and there's no cure. We turn to stem cell research to find regenerative therapy. There are two different types of stem cell transplantation. There is allogenic, which means it's taken from a donor. There are a couple of issues, one being that there's a donor shortage. Two, embryonic stem cells come from aborted fetuses, and that's an ethical concern for us. The rejection as well, they are foreign objects to our bodies, therefore our immune system would probably reject them. Autologous stem cells can be taken from the individual's fat cells, blood cells, bone marrow cells. Our research reviews clinical trials that will be safe and that will produce benefits that are sustainable, as well as to improve the quality of life of people living with type 1 diabetes. We are doing this by examining clinical trials that involve over 800 individuals living with type 1 diabetes. Age ranges are going to be 12 to 52 years old. Information that we have gathered is going to be the type of stem cell that was utilized, the duration of the study, if there were any adverse reactions, what parameters were used to assess the study. Then we want to discuss the condition of the patients after treatment. What are stem cells? They start off without any type of specialization, and then they differentiate and they become any type of cell that is needed. So what are the types of stem cells? So we'll see here in this diagram, they are embryonic stem cells, which are becoming all the different types of cells that are needed. If we look at adult stem cells, tissue specific, you're going to see umbilical cord stem cells, amniotic fluid stem cells, fat cells, and bone marrow cells. 6 stages of stem cell therapy. In this diagram, we have stem cells treated with synthetic growth factor. Having been harvested, now they are incubated. Cells are kept at a suitable temperature, they're frozen, and they are cultivated so that they can develop. Those stem cells are either returned by a drip, or if we were to take those cells and inject them into the heart, they would become heart cells. If we inject them into the brain, they're going to become brain cells. If you needed beta cells, you are going to inject the pancreas because the pancreas is what produces those insulin-producing beta cells. In this table, we're going to see basic information on patients in study. Here you'll note these are all type 1 diabetes patients, and then some studies actually list controls. Country of origin, Brazil was the first country to consider autologous stem cell transplantation in 2003 under adverse effects of study. While most of the studies produced no adverse reactions, the very first study, which was conducted in Brazil, lists that there were some complications, and that's why it's so important to continue stem cell research, because as our knowledge increases and improves, we are able to gain a better understanding to have a foresight to combat any reactions or side effects that may occur from the treatment. After looking at the country, you'll also notice that there's a duration of study from over 6 months all the way to 58 months. So this will show that the research that has been conducted is taking into account the sustainability and long-term impact and effects of these treatments. The studies incorporated individuals that were 12 years old, 13 years old, all the way to 52 years old, which is realistic because right now, type 1 diabetes is one of the leading autoimmune diseases. Types of stem cells that were used. We'll see mesenchymal stromal cells, bone marrow stem cells, autologous hematopoietic stem cell. Here we are going to look at the results of the clinical trial. You're going to notice parameters assessed, and there are parameters that are overlapping and that they have in common. Type 1 diabetes has low levels of insulin and C-peptide. Months after the studies were concluded, it showed a sustained improvement in levels of insulin, C-peptide levels, HbA1c levels, and most, if not all, participants in the studies developed an insulin independence. The types of stem cells that were used, it is a potentially good source for regenerative treatment. Our future plans are to gain a greater understanding on how sustainable regenerative stem cell treatments are for type 1 diabetes and also other diseases that presently have no cure. Serious life-altering injuries, such as ones associated with the spinal cord. I would like to thank Professor Chowdhury for being my mentor and helping me gain a greater understanding of stem cell research and type 1 diabetes. Thank you, Professor Yoel Rodriguez, for leading the CUNY Research Scholar Program seminars and allowing me to satisfy my scientific hunger on a weekly basis with these presentations that not only educate but inspire. Last but certainly not least, we have here our references. Thank you.